Now we are going to start installing forces involved in filtration while we are discussing about the function of kidney. So there are certain forces in the filtration. What forces favor and what forces oppose in the filtration of the blood through the kidney. And I have mentioned it before that kidney filtration is continuous 24 hours for life, and 20% of the cardiac output is filtered from the kidney, and it will remove all the toxic material, the metabolites which is remain in the blood after the metabolism. That is also an electrolytes, sodium, potassium, magnesium, which is not required for the body, or it retain or excrete out. Glomerular filtration. Glomerulus is a bunch of capillaries present in the Bowman capsule, and the blood comes in through those capillaries into the glomerulus Bowman capsule, and then this is filtered through the kidney tubules. Mechanism: bulk flow, direction of the movement from glomerulus capillaries to the capsule's space. Drive forces: pressure gradient, net filtration pressure, NFP. Type of the pressure: favoring forces, capillary blood pressure; opposite forces, blood colloidal osmotic pressures, COP, and capsule pressure. Glomerular filtration rate: amount of the filtration produced in the kidney each minute, 125 ml per minute. It means 180 liter per day. 180 liter means 18 bottle of filtered water. It is available from the Nestle's about 19 liter bottle. So make it 10 bottles, it will become 190 liter, and if you reduce out of that to 10 liter, then it will be 180 liter. 180 liter in 24 hours. Blood is continuously filtered and purified through the kidney. And if you go on the per minute, it is about 125 ml per minute. Factors that alter filtration pressure: these includes, means effect on the filtration: increase renal blood pressure, increase GFR. As much as the blood is available in the kidney, as much as glomerular filtration rate will be higher. Decrease plasma protein, increase GFR, causes edema. If there is a less protein available in the body, in the blood, then increase the GFR. It means it will filter with more speed and causes edema. Hemorrhage, bleeding, decrease capillary pressure, decrease GFR. Capsular pressure, capsules of the kidney. GFR regulation, adjusting blood flow. GFR is regulated by three mechanisms: renal autoregulation, means kidney autoregulation; neuronal regulation through the nervous system; hormonal regulation through the hormone controlled by the pituitary gland. All three mechanisms adjust renal blood pressure and resulting blood flow. Renal autoregulation. What is renal? ERPF, experimental renal plasma flow. GFR filtration rate. Mechanism. Myogenic mechanism means myo means muscles. Myogenic mechanism means how the muscles function of the kidney. Tubuloglomerular feedback means tubules, renal tubules, which is ascending, descending, and leukotriene, and the glomerulus, bunch of the vessels present in the Bowman capsule. Feedback, how they receive the blood. As much as they receive the blood, as much as they filter. Myogenic mechanism of the autoregulations. Efferent arterioles, glomerulus, efferent arterioles. Means blood comes from the efferent arterioles, then go to the glomerulus, bunch of the capillaries in the Bowman capsule, and then it will come out of that after filtration, and we call it efferent arteriole. Partial Oxygen pressure, BP increase, GFR decrease. If RBF, renal blood flow will decrease, then GFR also decrease. BP decrease, increase glomerular concentration, and increase the GFR, increase the renal blood flow. Blood flow, capillary pressure, flow resistance. Glomerular feedback mechanism as it is shown over here. Arterial pressure, glomerular capsule pressure, GFR, tubular flow, muscular dense juxtamedullary stimulation, vasoactive secretion, arterial vasoconstriction, arterial blood flow, glomerular capsule blood flow. This is all effect on the feedback. And what about, this was the auto regulation, and now it will come to neural regulation, which is controlled by the brain. Sympathetic nerve fibers in a way afferent and efferent arteries means that the blood comes into the Bowman capsule, glomerulus, and which leaves the glomerulus. Both have nerve innervations from the sympathetic nerve fibers. Normally, sympathetic stimulation is low but can increase during hemorrhage and exercise. The sympathetic system can be increases stimulated. Normally, sympathetic stimulation is low but can increase during hemorrhage and exercise. If there is a hemorrhage, bleeding, or exercise, then you can stimulate the sympathetic nervous system. Hormonal regulation of GFR. Angiotensin 2, supported vasoconstriction, constrictor, reduce GFR, glomerular filtration rate will be reduced through the hormonal regulation in which the angiotensin 2 is playing a major role. Arterial natriuretic peptides increase GFR by relaxing the afferent arteriole endothelium prostaglandin E2. Measuring GFR 125 ml per minute, 180 liter per day. Plasma clearance, the amount of the kind of substance present in the urine. The substance filtrate but neither reabsorbed nor segregated. If plasma concentration is 3 mg per liter, then 3 mg per liter into 180 days is equal to 540 mg per day. Known, unknown, and known. Reabsorption secretion, concept of reabsorption and secretion. Efferent arterioles, where it leave the Bowman capsules, then afferent arteriole, a place from where it entered to the glomerulus, Bowman capsule. 20% of the volume filtered, 19% of the fluid is reabsorbed. 19% at the tubular ends, it will be absorbed of the distal end. This is some of absorption and excretion rate of substances by the kidney. Two pathways of the absorption. One is lumen cell, then transcellular pathway, plasma, paracellular transport. Mechanism of transport. Primary active transport, secondary active transport, pinocytosis, and passive transport. So there are four types of mechanism of transport. One is a primary active transport. Second is secondary active transport. Third is pinocytosis, and fourth is passive transport. Primary, what is primary transport? Primary active transport means in the liver, sodium channel diffused and enter into the cell, tubular, then ADP, adenine diphosphate will convert into adenine diphosphate and produce energy, needs energy. Sodium, lateral space, basolateral sodium, potassium, aqueous carrier, and active transport will take place here. Secondary active transport, tubular lumen, tubular cells, co-transport system, out and in sodium, glucose out and in. In the same way, counter transport, antipode out in sodium and hydrogen from in to out. Counter transport will move one moiety, that is hydrogen, in the opposite direction to sodium. Co-transporter will move one moiety of, that is glucose, in the same direction as sodium. So one place H is replaced by the sodium and glucose is replaced by the sodium in another place. Passive transport, peritubular capillary blood bulk. It starts from the filtration, lumen, paracellular path, transcellular path, then solutes, active, passive diffusion, water, period, osmosis, and then it will transfer as a bulk flow, excretion on the right hand side, and reabsorption at the tubules. Pinocytosis, proximal tubules, reabsorb large molecules such as the protein, transportation of the sodium, water, and chloride. The proximal tubules include proximal convoluted tubule, thick descending segment of the loop of Henle. In proximal tubule, reabsorb about 65% of the filtered sodium, chlorine, bicarbonate, potassium, and essentially all the filtered glucose and amino acids. Secrets or organic acid based and hydrogen oil into the tubular lumen. Actually, most of the electrolytes are reabsorbed in the body because that is the requirement of the body for muscle contraction, for cardiac function, for creating action potential in the neuron. We need sodium and chlorine. So, reabsorption in proximal tubule, the sodium potassium ATPase, major source for reabsorption of the sodium and chlorine and water, means this is reabsorbed into the circulation, sodium, chlorine and water. In the first half of the proximal tubule, it is showing the location of the proximal tubule. Sodium is reabsorbed by the co-transport along with the glucose, amino acids and other solutes. Bicarbonate is preferentially reabsorbed with the secretion of hydrogen. Chlorine is not reabsorbed. In the second half of the proximal tubule, sodium reabsorbed mainly with the chlorine ion, use sodium chloride. So its concentration of the chlorine at the second half of the proximal tubule around 140 milligram per liter. Interstitial fluid about 105 milligram equivalent per liter. The higher chlorine concentration favor the diffusion of this oil. Sodium is passively reabsorbed down the electronic gradient. Sodium and water transport in loop of Henle. Loop of Henle is a turning, which is shown over here. This is loop of Henle. Constituent of the loop, the thin descending segment, the thin ascending segment, the thick ascending segment. Sodium and water transport in the loop of Henle, the descending loop of the Henle. High permeability to water and moderate permeability to most solutes. Has few mitochondria and little or not active reabsorption. Sodium and water transport in the loop of Henle, thick ascending loop of Henle, as shown in the figure. Reabsorption, about 25% of the filtrate load of the sodium, chlorine and potassium. Large amount of the calcium, bicarbonate and magnesium, they are all reabsorbed. Secretes hydrogen ion into the tubules. Mechanism of sodium, chlorine, potassium transport in the thick ascending loop of Henle. You can see over here. Tubular lumen, sodium, magnesium, calcium, then sodium, to chlorine ion, then potassium. And paracellular diffusion is taking place. Sodium goes out and potassium comes in the presence of ATP, because this is an energy packet for this pump to throw sodium out and potassium in the cell. In the same way, the chlorine comes out and the potassium will also comes out both sides. Glucose reabsorbed along with sodium in the early portion of the proximal tubule by secondary active transport. Essentially, all of the glucose is reabsorbed and no more than a few milligrams appear in the urine for 24 hours. The amount reabsorbed is proportionate to the amount filtered. When the transport maximum of glucose is exceed, the amount of glucose in the urine rises. The PMG is also about 375 milligrams per minute in men and 300 milligrams per minute in women. The renal threshold for glucose. The plasma level at which the glucose first appears in the urine, 200 milligrams per deciliter of the arterial plasma and 180 milligrams of venous blood. Hydrogen secretion and bicarbonate reabsorption. Hydrogen secretion through the secondary active transport, mainly at the proximal tubule, lopophenol, and early distal tubule. More than 90% of the bicarbonate is reabsorbed in this manner. This is a figure of the secondary active transport. This is how the bicarbonate is formed. It will the carbon dioxide attach with the water, the sodium, the bicarbonate, carbonic acid, and then it will H2CO3 is a bicarbonate, which is from RUBB, the combination of one hydrogen in the cell, SCO3 plus H, then H2CO3, carbonic anhydrase, then again it will break into water and carbon dioxide excrete out, exhale out, and one carbon dioxide is also made by the bicarbonate on the right hand side, hydrogen secretion bicarbonate reabsorption. Primary active transport of hydrogen, beginning in the late distal tubule and continuous through the remainder of the tubular system, occur at the lumen membrane of the tubular cell, transported directly by a specific protein, a hydrogen transporting adipase proton pump. We will continue in the next lecture further. So you have to understand whatever we have discussed till now.